Thank you for the invitation and good afternoon, everyone. Thank you for joining this TechExchange session. My name is Peter Lennon and I'm with MBF Bioscience. Today I'm going to be presenting the SLICE light sheet microscope system, which is a benchtop light sheet designed to make high-resolution 3D imaging of clear tissues more accessible to research laboratories. Light sheet microscopy has become a powerful tool for volumetric imaging of large biological specimens, especially in areas like neuroscience, developmental biology, and oncology. However, many traditional light sheet systems require significant infrastructure, complex alignment procedures, and a substantial financial investment. So the goal behind SLICE was to rethink how this technology could be delivered in a more practical and accessible format. By combining projected light sheet microscopy with an integrated computational pipeline, SLICE enables high resolution of clear tissues in a compact, easy-to-use platform. So today I'm going to briefly introduce the imaging technology behind SLICE, walk through the system workflow, and highlight a few applications where this approach is enabling insights into volumetric imaging experiments. So in general, light sheet microscopy works by illuminating the sample with a thin sheet of light that intersects the detection objective focal plane. This allows us to image an entire optical plane at once while minimizing photobleaching and out-of-focus light. This technique has become extremely powerful for imaging large cleared samples, including whole organs and even entire brains. However, traditional light sheet systems often require large optical tables, complex alignment procedures, specialized expertise to actually maintain. That complexity, cost, and even the cost of traditional commercial light sheet systems, as well as fragmented workflows, has really limited access to this technology for a lot of labs. The goal of SLICE is to remove those barriers so even non-experts can generate quantifiable 3D image data sets like you see here. There are many different types of light sheet arrangements available: open top light sheet theta microscopy, single objective light sheet microscopy, actually swept light sheet microscopy, and the list goes on. Today we're going to focus on a new cutting edge technique called PLSM, that's projected light sheet microscopy. SLICE is the next iteration of a system that was detailed in a peer-reviewed published paper by Dr. Raju Tomar at Columbia University. MBF Bioscience and longtime collaborator Dr. Tomar designed the SLICE system as a high-performance light sheet microscope in this compact, cost-effective platform, dramatically reducing mechanical, optical, and electronic complexity while lowering the overall system cost by orders of magnitude. As you can see here, this is a simple representation of the illumination light path and the detection pathway. In short, we wanted to bring light sheet to the masses and provide a simplified pipeline from acquisition to publication-ready, quantifiable data. So how do we simplify light sheet microscopy while still maintaining high performance? Well, I would say probably the key enabling technology in the SLICE platform is the concept of this projected light sheet microscopy. In a traditional light sheet system, the illumination sheet is often generated using specialized optics, such as a cylindrical lens or scanned Gaussian beams. And these configurations often require careful alignment and mechanical stability and utilize very expensive laser systems. In PLSM, the light sheet is generated through projected illumination patterns rather than fixed optical shaping elements. This approach allows the geometry of the light sheet, such as its thickness and position, to be controlled digitally by there's a built-in MEMS device in the light engine that we're actually using. Because the illumination pattern is generated computationally, the system becomes far simpler optically and much more robust mechanically. This innovation is one of the key reasons the SLICE platform can be implemented as a compact benchtop system rather than a larger custom type of optical setup. The result is light sheet microscopy in a very compact, cost-effective design. Building on the idea of projected light sheet technology, the SLICE system brings an entire workflow together. Here we see an example of the complete system. And no, your eyes are not deceiving you. The monitor we supply with the system is actually larger than the light sheet microscope itself. This is truly a benchtop type setup. This system has a dual opposing projected light sheet configuration, which improves illumination uniformity and penetration through clear tissues. The system also incorporates the concept of dynamic refractive shift correction. So it can accommodate multiple clearing protocols without any hardware changes or complex alignments. On the computational side, SLICE includes an end-to-end pipeline for automated acquisition, tile stitching, post-processing such as flat field correction, destriping, deconvolution, and high-performance 3D visualization, all within the same Bright Slice software environment. So this combination means researchers can go from intact clear tissue to fully visualized 3D data sets without needing any type of specialized optical systems or extensive computational expertise. Importantly, the entire system is available turnkey for under $100,000, which makes high-resolution volumetric imaging accessible to labs that would otherwise be excluded due to cost or complexity. And we actually include with the system itself, shown here, a 3-month free trial to our complete light sheet suite of analysis products for quantification of the data generated by the SLICE system. In developing the SLICE, we made several key improvements over the original PLSM design by the Tomer Lab to enhance its speed, precision, and image quality. We implemented a proprietary light engine for faster scanning and integrated a rapid sample positioning using a precision piezo XYZ stage. This allows us to acquire larger volumes more quickly without sacrificing resolution. The system also features improved spectral separation using a motorized emission filter wheel, enabling seamless acquisition of multi-channel images with a very user-friendly built-in workflow. Other optical design improvements extend the high image quality across a larger field of view while using a SCMOS backside illuminated camera that enhances sensitivity for low fluorescence signal type imaging. Finally, building on some more recent work from Dr. Tomer's lab, there is actually a 2025 preprint right now, we implemented a corrective optic in the detection pathway which further improves image uniformity and reduces aberrations, particularly for large clear tissues. Together, these enhancements make SLICE faster, more sensitive, and more reliable for high-resolution imaging. This achievement was also recognized with the 2025 Microscopy Today Innovation Award for its impact on advancing light sheet microscopy. The design changes that I just mentioned to the original PLSM enable SLICE to have the following characteristics. So on the illumination side, the platform supports 3 excitation channels, a 455, a 520, and a 640 nanometer laser for excitation. These wavelengths align with many common fluorophores used for imaging. The system uses a dual-arm light sheet illumination, producing a light sheet that's approximately 5 to 8 microns thick, depending on the parameters, how you set it up. And across the field of view, that's roughly 1.2 by about 0.75 millimeters using the standard 10x objective. On the detection side, the system supports either a 5x or a 10x objective, providing lateral resolution down to approximately 3/4 micron, 0.75 microns. So here you can see the difference between the field of view of our typical 10x objective or 5x objective, depending on our configuration. Samples are mounted in a cuvette configuration. So here you can see an example of the cuvette, and they magnetically lock into place on the piezo stage here. We have a scanning range of about 20 by 20 by 18 millimeters in size, which is sufficient for many clear tissue preparations, including whole mouse brains and even large organ sections. The other advantage of this cuvette-based sample mounting is that it reduces the required volume of clearing or refractive index media needed. So it lowers the overall reagent costs, operating costs of the system, while maintaining optimal imaging conditions. You do not need to fill this entire reservoir with your media. You only need to fill the small cuvette with the clearing media. And we do support a wide range of clearing methods, from aqueous to solvent-based protocols like iDisco, Clarity, cubic, binary, BAV, and this is all enabled by that dynamic refractive shift correction that I mentioned earlier. I did want to briefly highlight some of the fluorescence detection capabilities of the system. As mentioned, the SLICE platform uses 3 excitation channels, which correspond to blue, green, red spectral ranges commonly used for biological imaging. These excitation wavelengths are paired with optimized emission filters that allow detection of many widely used fluorophores. For example, the blue channel, we can image GFP, CFP, Alexa Fluor 48, which commonly used in viral labeling, reporter mouse lines. The green channel supports dyes such as Alexa 546, Alexa 555, Sci3, TD Tomato, which are frequently used for immunolabeling and neuronal tracing. And finally, the red channel enables imaging of far-red fluorophores such as Alexa 647, Sci5, TOPRO, which are particularly used for nuclear staining and deep tissue imaging in clear samples. Well, I have the slide up here. You can see an example of one of the data sets we acquired on SLICE, kind of playing in the background there. That's a projection, a slice projection going through multiple slices of a mouse brain cortex, basically acquired with our 10x objective. And the sample is GFP with secondary Alexa Fluor 488 amplification. I did want to touch in more detail about what I mentioned earlier with the dynamic refractive index correction. This is a really unique and powerful feature of the SLICE microscope. So in general, an important challenge in clear tissue light sheet imaging is refractive index variability. Different protocols, such as cubic, iDisco, Clarity, they all operate different refractive indices, typically ranging anywhere from 1.33 up to 1.56. When the refractive index of a sample medium changes, the optical path through the system changes as well. The result is that the light sheet can shift relative to the focal plane of the detection objective, and that's going to degrade your optical sectioning and reduce image quality. SLICE uses this idea of dynamic index correction, enabling automatic real-time compensation refractive index variability through the sample without requiring any manual optical realignments or changes to the hardware. To be clear, SLICE is doing this during the acquisition or while imaging live on the microscope. And this is all done through software-controlled optical offsets, meaning there's no hardware modifications, objective changes, or system reconfigurations, changes in the refractive index media. No hardware changes whatsoever are required when moving between different protocols. So as you can see here, on the left, we have a field of view tissue sample. This is an iDisco sample. So RI is roughly about 1.56. And this is imaged over multiple millimeters. And as you can see, when you start out on top, the cells are in focus, but as you go deeper into the tissue on the left, they go further and further out of focus. Whereas with the dynamic refractive index correction implemented, you can see the cell bodies actually stay nicely focused through the entirety of the material. So in practice, this allows researchers to image samples cleared with a wide range of methods while maintaining consistent optical sectioning and image quality without needing to retune the microscope at all. Here's just another example. This is Clarity. So now RI roughly 1.46. Again, multiple millimeters going through. You can see quickly on the left, the cell bodies and fibers lose focus. But on the right, we maintain crisp focus throughout the entire image volume. And again, no changes to the system whatsoever. We're simply swapping cuvettes, our sample, into the system without making any changes at all. Effectively, what you're doing is you're creating, you can think of it like a focus map. You go to the top of your image, you focus the microscope. You go to the bottom of your image, you focus your microscope using the digital offsets built into the system. And then we linearly interpolate between those 2 points. So here I'd like to show a few representative data sets acquired with the SLICE system to illustrate the range of biological applications. Starting in the top left is a FOSTRAP mouse brain expressing TD tomato, cleared using cubic R protocol. FOSTRAP enables targeted recombination in neurons that were active during a specific behavioral event, allowing researchers to map activity-dependent neural populations across the entire brain. In the top right, we have a mouse embryo at embryonic day 14, where neurons are labeled with size 3 and shown in orange with some tissue autofluorescence in cyan. The sample was cleared using iDisco, demonstrating the system's ability to visualize developing neuronal populations in intact embryos. Bottom left, we move to human tissue. This data set shows vascular imaging in neurotypical postmortem human brain sample processed using the Chanel clearing method. Imaging intact vascular networks at this scale enables quantitative studies of vascular architecture and pathology. The bottom center, we see zebrafish larvae brain at day 6, showing synaptic density in cyan, and GAD65 enzymes in magenta. This provides insight into neural circuits during development. And finally, on the bottom right, we have the brain of Astanax mexicanus, the Mexican cavefish, cleared with iDisco and labeled with TOPRO. Nuclear staining, this enables mapping of brain organization in the cavefish brain. Together, these data sets highlight how SLICE can be used to image whole organs and organisms, enabling applications ranging from neuroscience, developmental biology, to vascular analysis and pathology. Acquiring large tissue data sets generates extremely large image volumes, so efficient data management and visualization are critical. To address this, SLICE platform integrates end-to-end computational pipeline through the BrightSlice software. The BrightSlice software is included with every SLICE system. It features everything you need to control the microscope with easy-to-use workflows that could be operated by novice users, 3D stitching and post-processing filters to make your multi-tie acquisition into one final seamless image, 2D and 3D visualization tools for inspecting and generating publication-ready images or movies, and also it includes efficient data storage and management pipelines to keep your image data safe and accessible. We utilize a JPEG 2000 file format that allows for image compression while reducing the potential to introduce artifacts. This can take a data set that's multi-terabytes and compress it to just a few gigabytes or less. Once these volumetric data sets are reconstructed, they can be integrated downstream with some of our analysis tools. SLICE feeds into these image analysis software very seamlessly. And as I mentioned, you get a 3-month free trial of these tools with the system itself. So just some examples, you can register brain images to the Allen Brain Atlas and detect cells by region using our NeuroInfo software to reconstruct neuronal morphology with NeuroLucida 360, analyze and reconstruct vasculature using VesselsLucida 360. It's just a few examples of what you can do. All these tools are available to explore during the free trial. And together, these tools provide an integrated workflow from sample imaging to quantitative anatomical analysis. So just kind of in summary, despite the tremendous value of light sheet microscopy for volumetric imaging, widespread adoption has historically been limited by several practical barriers. Traditional light sheet systems can be extremely expensive, often costing several $100,000. They frequently require specialized infrastructure and expert operation. Again, the goal of SLICE platform is to lower those barriers of entry and make high-resolution, clear tissue imaging more accessible to a broader research community. Slice is designed as a compact benchtop system, providing the complete light sheet imaging solution at a cost significantly lower than most traditional or commercial systems. At the same time, the platform integrates automated acquisition, data processing, and the dynamic refractive index correction, allowing researchers to image samples cleared with different protocols without requiring any changes or complex optical adjustments. The result is a system that brings high resolution, volumetric imaging within the reach of many more laboratories, enabling applications across neuroscience, developmental biology, cancer research, tissue pathology. So with that, it's a lot of words there. I'd like to thank you for your attention, and I'd be happy to take some questions.